Hi, welcome to this chapter where we will be focusing on one structure that you were already introduced in our last module. But this time we will dive in with a lot more detail. So how about we get going? So there is a saying in my native language and a few other languages of the Nordic area which translates as something along the lines a dear child has many names. I guess that a good analog would be that Eskimos have a lot of words for snow. Basically, that means that if something is important or popular, this thing tends to have various different names that are used in different contexts. And the same is true also for the topic of this module, plasma membrane. And of course, we often know these as cell membranes. Just like we had a look of these in our last video, right? So there are quite a few other names or descriptions that you may see when we discuss about this structure. But the big thing for us to note is, after all this, that we need to know both the structure and function of this. As you will notice later in your studies in biology, structure, so anatomy, often explains the function, which can be referred as physiology. And of course, the other way around. Your textbook compares plasma membrane to the Grand Central Station. Despite all the hustle and bustle of the Central Station, it functions with a very high level of organization. In fact, people, their luggage, and other objects move from one location to another. They may cross certain boundaries or may be contained within these boundaries, such as various barriers, walls, and other structures that separate lines to the ticketing office, platforms, and so on. And of course, the passengers and other things traveling through this station move at a constant flow as part of a larger activity. This is an analog to plasma membrane, which has many functions, including movement within the cell and across the boundaries in various processes of the intracellular and intercellular activities. I guess that the big take-home message here is that the cell membrane is very active, dynamic, and constantly moving. So diagrams like this that you often see are misleading in a sense that the plasma membrane is not at all stationary or still, like these may end up leading you to think. Instead, they are very dynamic and changing, with parts within it moving and changing locations and various materials passing through, while other materials are not able to pass through these. And I should say here that much of this module's content is what we have already covered. And on the series of videos, we will be revisiting and expanding on these. I guess that the fact that this chapter is partially repetition just highlights how important these concepts really are for us. So let's look at some of the essential features of plasma membrane. And as the first one, I want to mention that it is plasma membrane that is in control of allowing certain materials to enter and exit the cell, as we saw, while other materials are prevented from crossing the membrane. The second thing to highlight about plasma membrane's properties is that they are able to change shape as needed. For example, here we see the cell membrane changing its shape as red blood cell squeezes through a capillary. Well, the next one that I would like to mention is that there are components within plasma membrane that allow us to recognize other cells. For example, whether a cell that we have came in contact with is one of our own cells or an invader. A cell that does not belong to our body. For example, diseases and pathogens are recognized in this way. At the same time, this can be sometimes problematic, such as with transplant rejection. So the last one that I have added here concerns a concept of transmission of messages. We have seen one example of this earlier with this example of a cell-to-cell communication in plasma modesmata of a plant cells. But there are other examples too. For example, hormones that bind into proteins that function as receptors. And one concept that we will see with these hormones is that hormones travel within the blood to everywhere in the body, but not every hormone affects every cell. So there must be a mechanism for controlling which cells are affected. And this is, of course, the receptors that are specific for that hormone and only found in cells that are intended to be affected by this hormone. What I would like to do next is to talk a little bit about the history of how we came up with our current understanding of the plasma membrane and what it is made of and what it does. What is amazing in my view is the fact that plasma membrane was first identified already in 1890s. Remember, Robert Hooke talked about these minute pockets. And these were, of course, the cells. And he was able to conclude that these cells were separated by plasma membranes. Of course, the terminology wasn't that well defined at that time. And it was in 1915 when it was identified that what this membrane is made of. And the main components of it were found out to be lipids and proteins. Imagine, already in 1950. Then the structure of plasma membrane was described, and a model for it given in 1935 by Davidson and Danieli. What they said was that a plasma membrane is like a railroad track, and they noted that it had two layers like sandwich. And if you think about it, it's not too far from the truth. Of course, Davidson and Danieli had this a little off in their proposed model, where they thought that the proteins were these tracks and lipids in between these. Of course, we know that it is actually the other way around. But with better microscopes and especially the availability of the transmission electron microscope, a new or improved model was formulated during 1950s to 1975. It was Singer and Nicholson who noted that this double layer fluid mosaic model could be explained by the structure and function of plasma membrane. So when I reflect on all this, I think that we have gotten really far with improving our understanding. But at the same time, I am amazed how good initial guess we had already early on, with much lesser resources to study the structure and function of plasma membrane. This is what makes me so excited to work in this field. Who knows what amazing things we are yet to discover. And let's have a look at this structure of a plasma membrane a little bit more, in more detail. We want to understand that where does the term fluid mosaic come from? Well, the term mosaic refers to the fact that it is made of many components. So we have phospholipids that we can see here, and proteins, of course, in fact, a lot of those. And we can also find cholesterol here and carbohydrate chains too. So just like a mosaic in arts is made of many different pieces which when put together form this full image, just like that we have plasma membrane that is made of many things which when put together form this elegant, well-functioning whole. So that's our mosaic aspect. How about the fluid nature of this membrane? What does the term fluid refer to? Well, the many components that make up the plasma membrane are in fact actually constantly moving about and changing their location and even position as far on which side of the membrane they are. This makes the plasma membrane a very dynamic and constantly changing, not only in its function, but also in its structure. So there is a lot going on at all the time. And as I have said before, what we see in these diagrams is only a snapshot in time, as normally the plasma membrane would never be this still. But that's not all at all. In addition, some materials are able to cross the plasma membrane, which also contributes to its fluid nature, while others, when they get blocked from crossing this membrane. So a lot going on in here that makes plasma membrane fluid in its characteristics too. And that of course gives us the description for the plasma membrane as being a fluid mosaic. And what we are going to do next is that we are going to look at a single phospholipid. As you remember, these were components that made most of the plasma membrane, and they were placed in it in these two layers that had the tails facing against each other and the heads facing outwards. But let's focus on a single phospholipid, as I said. So let's have a look at this enlarged diagram of one. And what we can do is that we can overlay the chemical structure of it here. And let's have a look at what it tells us. So here is the chemical structure of a single phospholipid. And here we see that it is made of a phosphate group, which forms the head part of the phospholipid, and a glycerol molecule, which is technically still considered as part of the head, and two fatty acid chains. These make up the tails of a phospholipid. Do notice also that there is also a double bond in one of the tails, causing a kink on its structure. This was of course unsaturated fatty acid chain, while the straight one with only single bonds is saturated. So this double bond causes a slight bend on the otherwise straight fatty acid tail, but only in another one of these. And now let's hide this chemical structure while leaving the components. And we see the head and tail parts that I have been talking about. And I know that this will be a review, but let's also mention that the head was hydrophilic, so it loves water. This means in practice that being attracted to the water, the head part of the phospholipid molecule always tries to arrange its position so that it is facing towards the water. While tail, which is hydrophobic, does what anyone with a phobia would do. It tries to get as far away from the source of phobia, so in this case water, as possible. So we see these tail parts always trying to arrange their position so that they are facing away from the water. Now, to recap, we have seen so far that tails will always aim to be avoiding the contact with water, and the heads will prefer to be in direct contact with it. But how about if we know that almost everything living, including human body, is made of water? We end up finding this water both inside the cells and outside them. So the big question ends up being now, where can the tails go to get away from water? Do you have an idea of what could work? And if you answered why not to have the tails facing towards each other, Well done, you are onto something. In fact, this is how plasma membranes are formed spontaneously in nature. And here we see one of the most common one of these, a sheet of phospholipids as a bilayer. And this is exactly what our cell membranes are like. But it doesn't have to be just flat sheets. In fact, these lipid bilayer spheres are quite common to break off from the cell membrane when we need to transport something large, say such a large molecule that it cannot fit through the membrane or even through the protein channels. So what happens is that the plasma membrane bilayer opens up and engulfs this protein, transporting it within a vesicle that it has formed in this way. And sometimes these phospholipids arrange so that there isn't even an empty space inside the sphere. It just ends up being a single layer. In fact, this is how many soaps work to remove fat and other molecules that we want gone. These balls surrounded by the material that is meant to be removed, separate it from the rest of the surface and take it away when washed away. But what is really important here for you as a takeaway message is that nature tries to form these bilayered phospholipid membranes spontaneously without any effort. And this is what gives rise to all membranes that we find in cells, both as cell membranes forming the outermost layer of the cell, but also as what ultimately forms all these compartments which we see on eukaryotic cells as the basis for the cell organelles. The next thing for us to do is to talk about proteins. And we have already discussed this earlier, especially in our last module. But here is a concept that I want to add to what we have learned so far. So often many proteins are fully embedded throughout the membrane, extending from one side to another side. These are known as integral proteins. Others instead are located on one periphery and do not extend all the way through the plasma membrane. These are therefore peripheral proteins. One thing to remember here though is that none of these proteins are stationary like these pictures may seem to suggest. Instead, they are constantly moving and even flipping on the side on which they might be located. Proteins can be also studied at various different levels of structure. Let's look at this concept. So here we see the primary structure, which is simply an amino acid chain. Remember how ribosomes used RNA template to build up a protein made of amino acids attached one after another one. Well, the secondary structure looks at this chemical structure, but considering it in a three-dimensional manner. Here we see examples of a pleated sheet and helical arrangements. And the tertiary structure takes again one step further away. We see the three-dimensional shape of an entire protein here. These are often shown as these globular or tube-like structures that are bent. But this is all good. But what's the practical application of all this? Well, in biology, the chemical structure defines also the physical shape of the protein. And this is an important concept to understand. Let's say that we have here a picture of Nicole Kidman. What does she have to do with proteins? Well, hair is made of protein, and you remember these levels on which we could look at the protein. We just covered these. And I want you to remember that when we change the chemical structure, we also will trigger changes in the physical shape of the protein. Well, Nicole Kidman does not have naturally this curly hair. She must have taken a perm to get it. And what the perm does is that it actually chemically alters the chemical structure of hair protein, which then results in a physically visible change in the protein's shape as well. So that's how Nicole Kidman relates to our discussion of proteins. Usually students hate it because it is so random, but I like to ask about this in the exam. So this next one should be a revision of what we just covered. Remember how some proteins expanded all the way through the plasma membrane? These were integral proteins. And some were located only on the inside or outside of the membrane, covering only part of its total thickness. These were peripheral proteins. Can we detect these in a more realistic drawing of a plasma membrane? Let's have a try. So here is an integral proteins. Remember all the way through for the full thickness of the plasma membrane. And here is peripheral. This is only one part way through, more on the only one side. So all this, what we have talked about so far, is anatomy of the cell membranes, so structures of these. And in our next video, we will move to look at the physiology, so the function of cell membranes. Thank you for having been part of this first video in this module. It will be really interesting content that we will cover in the next one.